Hello, dear children. Welcome back. I'm Dr. Jisha Joe. In this session, we shall discuss about ribosomes, microbodies, and cytoskeleton. First, let us move on to what are ribosomes. Ribosomes are the smallest cell organelles. They are composed of RNA and proteins. They are small granular structures and are not bounded by any membrane. Okay, they are the smallest cell organelles. They are composed of RNA and proteins. They are granular structures which are not bounded by any membrane. And we know ribosomes are the sites of protein synthesis. They are the regions where protein synthesis occurs. Ribosomes were first discovered by Robinson and Brown. Robinson and Brown discovered the ribosomes. And it was George Palade who coined the term ribosomes. The term ribosome was coined by George Palade, and it was Robinson and Brown who discovered the ribosomes. Okay, so once again, ribosomes are the sites of protein synthesis. They are small granular structures which are not bounded by any membrane. They are the smallest cell organelles and are composed of RNA and proteins. Now, where are these ribosomes present in the cell? Do you remember we have already discussed about this before when we have discussed about the endoplasmic reticulum. We can see the ribosomes attached to the endoplasmic reticulum, and also ribosomes can be seen freely occurring in the cytoplasm. So, where are the ribosomes present? Ribosomes may be seen freely in the cytoplasm or may be seen attached to the endoplasmic reticulum. Whether they are seen freely in the cytoplasm or whether they are seen attached to the endoplasmic reticulum, the function of the ribosomes is the synthesis of proteins. Now, ribosomes are present in both prokaryotic and eukaryotic cells. And but there is a difference in the type of ribosomes present in these two types of cells, and that is how we can say that the ribosomes are of two types. Okay, the 70S ribosomes which are present in prokaryotic cells, and the 80S ribosomes which are present in the eukaryotic cells. The S stands for Swedberg unit. Swedberg unit. It is Swedberg unit. It is the sedimentation coefficient. And what is sedimentation coefficient? It is the velocity with which a molecule sediments through a solution. The velocity with which a molecule sediments through a solution is called as the sedimentation coefficient. Okay. And this sedimentation coefficient depends upon the molecular weight of the molecule and as well as its shape. The shape and the molecular weight of the molecule determines the sedimentation coefficient. So, ribosomes are of two types, the 70S ribosomes which are seen in prokaryotic cells and the 80S ribosomes which are seen in eukaryotic cells. Now, if you look at each ribosome, every ribosome will have two subunits. Two subunits together form a single ribosome. There is a larger subunit and a smaller subunit. Whether it is the 70S or the 80S ribosome, it will have two subunits, a larger subunit and a smaller subunit. The 70S ribosomes will have the two subunits 50S and 30S. The 50S subunit is the largest subunit and 30S is the smaller subunit, and the 80S ribosomes which are seen in eukaryotic cells also have two subunits. They are the 60S and 40S subunits. So we have discussed that each ribosome, whether it is the 70S or the 80S ribosomes, will have the two subunits, a larger subunit and a smaller subunit, and the two subunits of a ribosome are joined together by magnesium ions. Magnesium ions join the two subunits of a ribosome. Next, we shall see what are microbodies. Microbodies are membrane-bound vesicles that are present inside the cell. They are seen both in plant cell as well as in animal cell. So, membrane-bound vesicles. Vesicles means they are spherical bodies which are bounded by a membrane. And the function of the microbodies is that they contain enzymes. Enzymes, we know enzymes are needed for various chemical reactions that are taking place in the cell. So it is these microbodies which contain the enzymes. So what are microbodies? Microbodies are membrane-bound vesicles that are present inside the cell. They are present in both plant cell as well as in animal cell, and the function of the vesicles is to store the enzymes. Next, we shall move on to cytoskeleton. As the term indicates, cytoskeleton means forms a skeletal framework for the cell. It is actually consisting of a network of filamentous proteinaceous structures that are seen in the cytoplasm. They act as a skeletal framework for the cell. Okay, it is a network of filamentous proteinaceous structures present inside the cytoplasm of the cell. And the function of the cytoskeleton is the three M's. They are concerned with motility. Motility means the movement. They are concerned with motility. Then mechanical support. They provide mechanical support to the cell. And also the last function, the third function, is the maintenance of shape of the cell. Maintenance of shape. So these are the functions of cytoskeleton. Okay, cytoskeleton is a network of filamentous proteinaceous structures present in the cytoplasm of the cell, and they are concerned with motility, mechanical support, and the maintenance of shape of the cell. There are three major classes of cytoskeleton. The first one is the microtubule. Okay, so three classes. The first is the microtubules. So microtubules, they consist of long, hollow, unbranched tubes. Long, hollow, unbranched tubes. And they have a diameter of 21 to 24 nanometers. Okay, they have a diameter, these hollow tubes have a diameter of 21 to 24 nanometers. And they are made up of a protein called tubulin. Tubulin is the type of protein that is present in microtubules. And where are they present? They are present in the cilia. Okay, they are present in cilia, in the flagella of eukaryotic cells, and in the centrioles you can see the microtubules. Okay, so that is with regard to the first class of cytoskeleton, which is the microtubules. Microtubules are formed of a protein called tubulin. They consist of long, hollow, unbranched tubes with a diameter of about 21 to 24 nanometers. And they are present in the cilia, flagella of eukaryotic cells, and in the centrioles. Now, the functions of microtubules. Microtubules are concerned with cell movement. As you can see in the case of the cilia, flagella and all, it helps in movement of the cell. And also, it is the microtubules that help in maintenance of the shape of the cell. The shape of the cell is maintained with the help of microtubules. So, that is the first class of cytoskeleton. Now coming to the second class of cytoskeleton, which is the intermediate filament. Intermediate filament. That is the second class of cytoskeleton, which is the intermediate filament. This intermediate filament has a diameter of 7 to 11 nanometers. Okay, 7 to 11 nanometer in diameter, and the function of intermediate filaments is the maintenance of the shape of the cell. Helps in maintaining the shape of the cell. So now moving on to the third class of cytoskeleton, they are the microfilaments. Microfilaments, they have a diameter of 7 nanometers. Okay, 7 nanometer in diameter, and they are of two types. Two types of microfilaments are there. They are two types. Okay, they are actin and myosin. Actin and myosin. And the function of these microfilaments is to help in the contraction of muscles. So it is actin and myosin microfilaments that help in contraction of the skeletal muscles. In non-muscular cells, the microfilaments give rigidity to the cell. Okay, so major function or the important function of microfilaments in skeletal muscles, it helps in the contraction of the muscles, and in non-muscular cells they give rigidity to the cell. So dear children, I hope this session is clear to you. If you have any doubts or suggestions, please ask your doubts and give your suggestions in the comment section. Thank you.